Welcome back. Now that we have seen what are enzymes and how they work by reducing the activation energy and they are specific, let us see some of the theories that aim to explain enzyme action. Welcome. So, what about enzyme action? So we have seen that enzymes interact with their substrates. To catalyze the reaction, we have to have the enzyme substrate complex. And one of the things we know that enzymes exhibit different types of specificity. One of the things we say is that enzymes are very specific to the substrate they can catalyze. However, we see different types of specificity. One of the specificity we see is a group specificity, where we can see a particular enzyme works on related substances that might have a common functional group. For example, alcohol dehydrogenase can work on ethanol and methanol. Both of them have the alcohol group. So that means this alcohol dehydrogenase does not recognize the ethanol or the methanol, it recognizes the OH group. So this is a type of specificity we refer to as group specificity. And we are looking at the functional group, which in this case is the alcohol group, the OH group. Okay. Another type of specificity we note about enzymes is the absolute specificity, where the enzyme will work on only one substrate, one specific substrate. A good example is an enzyme which we call glucokinase, which is very specific to glucose and can phosphorylate it to glucose 6-phosphate. This can be highlighted by another enzyme which can also phosphorylate glucose, which is referred to as hexokinase. This hexokinase is not absolutely specific to glucose. It can also phosphorylate galactose. It can also phosphorylate fructose. So long as it is a hexose, it will phosphorylate it. However, glucokinase is absolutely specific for glucose and it will not phosphorylate fructose or galactose. So this is a very good example of absolute specificity. Okay, so we can see this is another type of specificity. Then we have enzymes that produce a specific product. Maybe we could have a reaction that is happening in the environment and we can have two forms of the product. Usually isomers, the product have similar structure, but the orientation of the molecules in space is different. However, what we see in biological enzymes, they are very specific in the product they produce. They'll produce one specific isomer. This is another specificity we tend to see with biological and catalysts or enzymes. And then we have the stereochemical specificity, where here only one isomer of the substrate will be acted upon. For example, we have seen we can either have L amino acids or D amino acids. We have seen an enzyme L amino acid oxidase that can only act on L amino acids. If we have the isomer D amino acids, this enzyme will not work on D amino acids. So we can say these types of specificity. Now that we know that the enzyme has to bind to the active site, these theories we are talking about are hypotheses to try and explain how do the structure of the enzyme relate to the structure of the substrate. So that's why we have these hypotheses. They try to explain the kind of different specificity we see in different enzymes. So the first hypothesis is referred to as the lock and key hypothesis, which was proposed by Emil Fischer in 1894. In his hypothesis, he says the structure of the substrate matches completely with the substructure of the enzyme, much like the key fits into a lock. Okay, and this would be very good at explaining the absolute specificity, such that when glucose react with when glucose is the substrate for glucokinase, it matches exactly just like a key fits into a lock. Okay, and it's very good at explaining absolute specificity. So that means in the lock and key hypothesis, there's no modification of either the enzyme and the substrate, much like the key. If you bring another key that looks similar to the key, but it's not for the lock, it will not open the lock. So that means here we are showing high specificity. However, This lock and key model has a challenge because we have seen other types of specificity like group specificity of alcohol dehydrogenase, and we also see a lot of flexibility in enzyme substrate reaction which the lock and key cannot explain. As we know, a lock has only one key. So we have seen some enzymes like hexokinase can bind structurally similar substrates, hexoses, or alcohol dehydrogenase, which can bind both methanol and ethanol. So the lock and key cannot really explain how this other enzyme works. So it ignores the conformational changes in enzymes which are observed in some of those reactions we see. So that's why we have a second hypothesis, the induced fit hypothesis. This induced fit hypothesis was proposed by Daniel Koshland in 1958. And in this hypothesis, it hypothesizes that the active site is flexible and it can undergo some conformational change to fit with the substrate, much like a glove. It's also called much like a glove. For example, when you see the glove, it has the shape of a hand, but it does not is not fitting to every hand, but the glove adapts to the hand that fits in. Here we can see the active site has a similar kind of shape, doesn't match exactly with the shape of the active site with the substrate. However, once the substrate bind, the active shape adapts the change to fit, just like the gloves fit into the hand of the wearer of the glove. So the induced fit hypothesis explains some of the things we see, like how hexokinase can phosphorylate several hexoses. And this adapting to the shape of the substrate ensures the enzyme the tighter binding between the enzyme and the substrate and better catalysis. So the induced fit hypothesis is the more widely accepted as it explains the flexibility of some of the enzymes we see. If we take a moment to compare the two models, we have seen the lock and key is a rigid hypothesis. There's a rigid fit, it's highly specific, and it only explains the absolute specificity. However, we have seen in the induced fit, the enzyme, the active site is flexible, so there's flexible binding, and we see that the enzyme adjusts to fit the substrate, and it explains both group and stereochemical specificity. So usually the induced fit is thought to better explain enzyme specificity than the lock and key hypothesis, although both of them have their place in explaining how substrate and enzyme bind. So thank you. For the key takeaway, we have seen the two models, the lock and key, which is simple but lacks flexibility, and the induced fit, which better describes enzyme action and adaptability. So we know enzyme function is very crucial for metabolism and drug development, and it's very important to understand how the enzymes interact with the substrate. Asanteni sana. See you in the next one.